I'm Mike McMaster from UCSF. I am a developmental biologist, not a toxicologist or an epidemiologist. This is the disclaimer part of my talk, I suppose. I also am not going to say too much about autism, although listening to Eric is very interesting. Like many of the points I hope to bring out in my talk, which is basically about how we might think about applying stem cells to predictive toxicology studies to replace some of the methods that are currently being used. I mean, what came out was 8 or 10 points he made specifically relate to exactly the same kind of, at least conceptual thinking I think we need to have related to how this is going to work. Environmental exposures, developmental exposures. Most of my stuff has been developmental toxicology, not sort of adult cells and environment, but hopefully these parallels will come out, even though I'm not going to specifically talk about autism. What I wanted to start out by doing here is trying to just sort of frame the discussion around what the current issues are in applying stem cell biology to understanding the effects of toxicological exposure, both in the developmental sense and in adult organisms. This slide is basically just meant to point out something that's been very well outlined. Actually, I highly recommend the report from the workshop of 2 years ago. It's great reading that all the things that I'm going to say today are pretty much outlined in much more detail in that report from the meeting that happened, I guess, about 18 months ago. Obviously, animal studies don't get us where we need to go. Very expensive, very slow. It's very interesting that many of the drug candidates, for example, have made it through animal studies, get to human clinical trials, and they fail. And of course, commercial interest, big pharma is very interested in finding a way to get these failure numbers down. And I think this might be something that stem cells could be useful for. In vitro assays, the current sort of ways people do these kind of studies involve immortalized cell lines, essentially call them cancer cell line cells that will just grow rampantly in a dish. Hyperproliferative, meaning that they're selected for their ability to divide, which is not really a normal characteristic of cells. So then when you hit them with a test compound, you can't really expect that the response you're going to get from those cells is really representative of what's truly happening in a physiologic context in an adult or developmental situation. I put down here these, as an example, these isoprotocols for cytotoxicity screening. If you go read these things, there are guidelines for using cell lines that you can get from ATCC, which is a sort of a bank of cell lines that's out there, that basically very specific protocols that the FDA looks to, for example. And the endpoints are the cells look terrible, the cells die. Right? That doesn't really begin to tease out mechanisms or get you anywhere closer to understanding the true toxicity or potential sequela of, for example, a drug or a toxin in the environment. The other thing that's possible, of course, is to use primary cells. Take a tissue, grow some cells out in a dish, expose those, and it's probably closer to the real situation. But the problem with that is that it's difficult to get cells like this all the time. You need fresh tissue all the time. The results are quite inconsistent. Again, this is all concepts that have been framed in this workshop report that I encourage you to read. So part of the reason we're here today is to talk about what the solutions to some of this might be. And I just sort of made this little outline. This first part here, it says in vitro HESC assays. And I'm making the differentiation here between developmental studies where you might study undifferentiated stem cells in the dish, which much of the work that we've done, as compared to what's probably more relevant to much of the toxicology that people are interested in, and also for drug development, actually looking at adult differentiated cell types. Take embryonic stem cells, differentiate them into liver cells or nerve cells or cardiomyocytes, then I will do the studies. Quite a different approach, or at least biologically, developmentally thinking, whether you're talking about developmental studies or say gestational exposures or how exposures might affect development in the way that Eric was talking about. I'm very interested in that. But it's a quite different thing to be talking about sort of high throughput assays where you might have cells in a dish that represent neurons, let's say, that you can then expose to pesticides or toxins and ask the question, what's the damage, what's the specific cellular mechanisms that are getting disrupted? It's also, I mean, when you think about stem cells in this context, you can't forget the idea that it's not just about in vitro or cells in the dish. There's also approaches that you can use where you sort of combine in vivo and in vitro. You can take embryonic stem cells, put them into a mouse. So you have human cells that are going to respond in a way presumably that other human cells might respond, but you then solve the problem of the lack of sort of physiologic environment, which of course you don't have in mice. Mice that have human immune systems that have been sort of grafted in there. An amazing thing. But you know, the potential then is to look at the response to, for example, myeloproliferative cells based on testing that you can do. The third thing I have on this list here is iPS cells, induced pluripotent stem cells, getting to the idea that as opposed to, you know, undifferentiated human embryonic stem cells, the potential exists to take cells from known genetic backgrounds, for example, family or a patient that you know has a particular condition, make induced pluripotent stem cells, then do the testing. It's very, what would you say, tantalizing way to get this whole idea of epigenetic versus genetic. What's the genetic background that is susceptible to a particular set of insults that are coming from the environment or from drugs that get prescribed by your physician. Okay, so that is sort of my way, take it or leave it, of sort of framing what I think I was asked to talk about today. So here specifically is what I'm going to do in the next 10 minutes or so. We've been doing some studies using embryonic stem cells on nicotine. And this is work that's funded by the TRDRP, the Tobacco-Related Disease Research Program. I think this is the state tobacco funding mechanism. I hope I got that right. Obviously, because maternal smoking has been associated with a bunch of things I'll tell you about in a second. So this is my way of just sort of giving you a sample of the kind of approaches you might do on using stem cells for predictive toxicology. You can sort of substitute your favorite molecule into this sort of workflow or experimental design I'm about to describe in order to get at whatever particular disease or thing you're interested in studying. Then a really quick description of a grant proposal that we have in the hopper now to look at BPA using effects of toxicity of BPA using stem cell biology. And then finally, just a few slides on, hopefully again, sort of framing the major issues, really stuff that's coming out of that workshop report. So let me just tell you a little bit about this nicotine study we've done. It's sort of ongoing. There's one paper that's come out from this. The rest of the stuff is still being analyzed. The general issue is this. So maternal smoking is associated with lots of bad things. I think in this room, sort of nobody smokes very much anymore in San Francisco and all that. I noticed I commute across the Bay Bridge every day. That's where people smoke, is in their cars. Because it's the only place left you can actually smoke. I don't think anybody would disagree with the fact that it's a bad thing to be doing in general, and particularly when you're pregnant, but pregnant women smoke, particularly in Europe. Low birth weight is very much associated with smoking. Delayed conception, cases of infertility have been reported to be related to smoking. Premature birth, sudden infant death, this very severe orophacial clefting that's a congenital defect that's been shown to be associated with maternal smoking.
Excuse me, is the infertility in the child that was exposed?
No, the infertility is in the mom, right? So it's not exactly results happening in the pregnancy with the mom. It's interesting you say that, though. No, just a brief aside. I mean, there's a whole literature developing now about your intrauterine programming of future adult health. And this comes from the work by a guy named Barker in the UK, who basically showed that during World War II, these English countryside or villages, whatever they were, that were blocked from the supply lines had a lot of starvation going on. And women who had malnutrition during pregnancy gave birth to a cohort of kids who had extremely, like 3 to 6-fold time increased risk of things like type 2 diabetes, coronary artery disease in their 50s. So intrauterine programming isn't just about having a baby with a birth defect or premature birth with all the bad things that happen with that. It's about programming your adult health all the way through through your reproductive years and beyond. So a lot of the work we did on this looked initially at placentation and sort of extra embryonic tissues that are affected by smoking, growth restriction is such a big deal that you can easily make a case that if your placenta isn't working right, you don't have good circulation, et cetera, et cetera. I'm not going to talk about that. If we then sort of switch to modeling nicotine effect in embryonic stem cells as a way to model some of the events or the insults that might be happening to the fetus itself, the early embryonic period that Eric was talking about, precisely the kind of developmental period that we're trying to model by using embryonic stem cells. So these next few slides are just some data from this work. I won't dwell on this stuff too much. If you take a colony of embryonic stem cells, that's what this is right here. It's sort of sitting on a feeder layer, nice, happy-looking cells, refractile little colony, and hit them with nicotine at different dosages. They basically start to look like hell. They develop these vacuoles, they lose their cell-cell contact, they're just not very happy cells. At a high dose here of 3.7 micromolar, you see that they no longer like to hang around with each other. They've sort of lost their ability to connect and spread out in the dish. I should just briefly mention, and this gets to a general point later about dosages, is that toxicology studies and pharmacokinetic studies have long been criticized for using in vitro studies hyperphysiologic or non-physiologic doses. So I think it's important in all these kind of studies to say that we're using environmentally relevant doses to show effects on cells, whether they be stem cells or anything else.
Just a quick question.
Yep.
Are these cells more sensitive than somatic cells? If you did the same experiment with fibroblasts or cancer cells,
Yes.
would you see?
That's a good question. That's a really good question. And I think the answer is yes, you would. There's evidence in the literature of fibroblast cell lines. Again, it's this cell line issue that I mentioned earlier, but you get the same kind of responses in those cells that you do in these embryonic stem cells. The dosage thing comes from some measurements of nicotine or cotinine concentration in cervical mucus, which goes as high as 6 micromolar, with the idea being that perhaps it's not so important to understand what blood levels might be, but it's actually the tissue level that becomes important in a physiologic way. Very simple little graph here just showing that I don't know if you can read this, it says percent attached cells. It's basically showing quantitatively now that in a dose response curve, the more nicotine the cells are exposed to, the more they basically fall off the dish or lose their contacts between each other. This is receptor mediated, so nicotine acts through acetylcholine receptor, nicotinic cholinergic receptor. And this drug right here, turbo-curare, is an antagonist of that receptor. It's basically just showing that here's the control situation, here's what happens if you hit them with nicotine. And if you hit them with nicotine and this drug that blocks the receptor, they come back to control levels, demonstrating that it's actually acting through that receptor. Basic sort of thing that you do in studies like this. We also showed that these sort of so-called stemness markers that are associated with embryonic stem cells in their sort of totipotent or proliferative state, OCT3/4 and TRA181 in this case, are altered excuse me, by exposure to nicotine. You can't really see it very well in this room, but these basically they're immunofluorescence pictures, but you're looking for red cells, basically. You can see here there's fewer red cells, there's less of this protein being made when it's exposed to nicotine. Same thing with this one here, TRA181. What this sort of the green panel below are showing is that it's not necessarily quantitative differences in those proteins as qualitative. What this body panel is showing is that the molecules are redistributed within the cell. So where they might exist in the cytoplasm or on the cell surface, after treatment with drugs, they redistribute to different cellular localizations, suggesting obviously that function is going to be different or disrupted or whatever you might find in your particular case. This is a measure of apoptosis, so-called programmed cell death. Cells have a very interesting pathway in them, which is sort of a suicide pathway, apoptosis it's called, where they get a particular stimulus and the cells commit suicide, for lack of a better word, grind up their DNA, die. Very, very different from sort of necrotic cell death, where the cells can die from their membranes being compromised or whatever. This apoptotic pathway is a very important thing in development. The example people always use is the webbing between your fingers, right? During development, there's a stage where you have this webbing between your fingers. Those cells, through whatever genetic program, become apoptotic and die off, and you get fingers like we have instead of duck feet, let's say. Apoptosis is a very important mechanism, and nicotine causes apoptosis in the cells. Another sort of mechanism you might want to follow if you're trying to figure out what the particular cellular and molecular mechanisms are stimulated by nicotine that result in all these bad things that I listed on the slide. So from there, so that stuff's all published. I should give credit to these people. This is published last year in Reproductive Toxicology. We've then gone on to sort of take a different approach. So now we can see phenotypically, morphologically, the drug is not good for these cells. Duh, right? It's not surprising that it messes them up. And we can start to point to particular mechanisms, but you really want to dissect it at a genetic or a much finer molecular way. We have then applied this microarray technology. It's a way to basically get a readout of all the genes in the cell and whether they're turned on or turned off, or relatively expressed at a higher or lower level compared to a control condition. Pretty common technology. Most people sort of understand this is going on. Affymetrix is making tons of money selling these chips where you can take your cells, you extract the RNA, you do a hybridization analysis, and you get tons and tons of data. Gene A is turned off relative to the control situation or turned on. This is just a quick little picture to show you how the experiment works. You grow your cells exposed to whatever molecule of interest. You extract the RNA, which is the readout of the DNA, and validate it, make sure your RNA looks good, put it in these chips, and you get these sort of heat maps that tell you in a relative way expression of all the genes. This is straight out of the Genome Project, right? Because now we can say all the genes. It's kind of confusing. We don't know what all the genes are, and if you look at a list like this, there'll be, what's an example? Rhomboid 5 homolog from Drosophila. Nobody really knows what that is functionally, but we know it's like a gene that somebody studied in a fruit fly. So all this stuff is in the annotated genome. But the point is you're able to survey all the genes and get candidates for understanding the molecular pathways induced by whatever insult we're talking about. This is just a quick list of what we found in one of these experiments. This is just 2 doses. Red means low, blue means high. I'm sorry, I have that backwards. Blue is low and red is high. So this is a lower dose of nicotine here, and we're showing that these genes are relatively increased if you treat it with nicotine. I won't get into any of the details of this, but you can then start to do pathway analysis and figure out which pathways, which groups of genes, which signaling mechanisms are activated or suppressed by a particular thing. This is just a summary of the data. There were 63 upregulated genes and 59 downregulated genes at this particular dose, and a different number at this particular dose, not particularly nicotine. One of our most intriguing hits was a gene called oral facial cleft candidate 1, upregulated in stem cells that have been exposed to nicotine. You put that along with the fact that this, granted rare but severe congenital defect, is increased in smoking women. We haven't proven cause and effect, but it sure is tantalizing to get somebody then to go study that molecule and try to follow up that pathway and find out if indeed some women, for whatever reason, maybe based on their genetic background, upregulate that gene related to smoking, and if it hits at a particular developmental stage, as Eric was pointing out, it could lead to these types of problems.
Excuse me, so this particular gene has been implicated as potentially being the cause of a genetic orophacial clefting defect.
That's right. That's right. So I don't want to drill down this too far, but mutations in that gene are associated with it. But presumably, some of those mutations could be loss of function or increased function mutations that are basically saying the same thing. You don't necessarily have to have a mutation. You just have to have an abnormal pattern of expression of that gene to give you the problem.
I'm wondering if people with autism have had their, like a large enough group, have been mapped that we are getting any kind of information.
I think the answer to that is no. I'm sort of no expert on this, but I think not. It's obviously a ripe area for this. I mean, I hesitate to go too far into this. I'm not an autism expert at all.
Okay.
But it seems to me that like all these kind of diseases, it's going to be a nature-nurture thing, right? Genetics and epigenetics. There's a genetic background that has susceptibility to autism or you name the disease. And then there's perhaps a certain set of developmental sequelae or environmental insults that then on that genetic background result in the problem. If you can understand that genetic background and the types of things that happen with all the genes going up and down related to pesticides or whatever it might be, that's absolutely where I'm suggesting we could go.
I think the commonly cited statistics now are that about 10% of the autism cases we seem to see, maybe 10 to 20, have actually a genetic component that we can identify, but there's no single gene that accounts for more than 1% of the cases. And practically all of the genes you find in normal controls too. So it's sort of, genetic analysis of autism has been amazingly fruitful, but there's also sort of this nagging question of it's been fruitful and maybe it's sort of done and it needs to go.
This is the
Okay,
problem
so
with all these kind of
be
things.
carried out further.
Right,
Okay.
exactly.
Right. Understanding the interactions between expression of all these genes, that's where all biology is. It's so complex and so difficult to tease out. That's the whole audience. So this is just a summary of pathway analysis. Then you can put all this data into the software that allows you to say, you know, if these 10 genes are up and these 10 genes are down, that means this particular pathway is active or inactive or better. Cell adhesion mechanisms are very much affected by nicotine exposure. It makes sense for what we see in the dish. It makes sense for the biology of what happens in these pregnancies. Apoptotic pathways are also active. Good correlation, almost a positive control for us. We see apoptosis in the dish. It's nice to see that we're detecting those genes in that pathway in the same way in our molecular analysis. Markers of pluripotency, same thing we saw it in a dish. It's good to see it at the molecular or mRNA level, cell survival signals, etc. So the idea then is that you use this sort of complicated and voluminous, actually, molecular data to develop hypotheses about what the mechanisms are that are actually causing all these kinds of things. I'm going to blast through these slides. I'm not going to get through to the details, but the point I want to make is that the key is to this, or one important concept of this is validation. You can get tons and tons of lists of genes that have gone up or down. You need to go back then and ask the question, is this really happening at the protein level? Is it relevant in a particular cell type, et cetera, et cetera? The worst thing you could do is take big piles of data like that and make grand sweeping statements without going into so-called validation of the results. This is just biochemical data validating this molecule atom TS17. This is the PCR data, this is the protein data proving it's actually doing what our microarray analysis told us it was probably doing. So this is just one slide to point to a study that I wrote up with Tracy. It's in the funding hopper right now, to basically do the same types of approaches, this time with BPA, bromophenol A. The study design is basically to treat the stem cells with BPA in environmentally relevant doses, measure the global changes in gene expression like I just described in comparison to estrogen. This gets to what Eric was saying too about environmental estrogens and endocrine disruptors. Many pesticides and other types of molecules in the environment are so-called endocrine disruptors, meaning they act like hormones. And we can all attest in this room, like estrogen, for example, is an extremely powerful hormone that does all sorts of things, good and bad, you know. And BPA is estrogenic, and so the idea of the study design is to say that if we're going to look at the effects of BPA on stem cells, let's find out which of those are related to the molecule itself, irrespective of its interaction with the estrogen receptor, which ones are actually pure estrogenic responses. And then in addition, we monitor their ability to differentiate. Can't get too stuck in the dish of molecules. You have to make sure it's actually sort of real life and things that you can point to as being important for what's already known about behavior of these cells. And then the whole pathway analysis thing. The idea being that maybe we can develop a phenotype for exposure. What's the molecular phenotype? What are the set of genes that are up and down after exposure to BPA or any, you name it, chemical or drug candidate? And if you then have that phenotype and know, you can develop tests. So So does this person have in their blood some readout that we can measure as a physiological response to exposure to whatever it is that we're interested in studying? And then, of course, can we develop data to inform public health guidelines? I meant to say in my introduction that I love coming to things like this because I think, I'm a developmental biologist, I'm not an epi person, I'm not a tox person, but I think it's that interface of disciplines like this where stuff gets done. I mean, that's where everything gets done. We can sit down and, sit in our lab and look at the effects on cells all day long and write papers and they go in the journals and we get promoted and all that. But what actually matters is getting this stuff out there and having people with completely different backgrounds, like I think is represented in this room. That's how things get done. Okay, so this is just the sort of hand-waving slide. Is this really going to work? Yes. The next few slides here are just sort of an outline of what I think are sort of the major issues to be thought about. If you want to apply embryonic stem cell techniques and biology and assays to these type of drug discovery workflows, or understanding developmental toxicities or environmental toxicities, these are kind of the main issues. And again, I point you to this workshop report. It's really the same types of issues that have been fleshed out much better there than I did on my slides here. So what stage of development is actually being modeled? Are we talking about a gestational exposure important that's going to cause a developmental defect? Or are we talking about an adult person with an adult nervous system being exposed to whatever it is and having an effect? And it's just sort of an experimental design, and you've got to be clear. Embryonic stem cells, as they exist in a dish in their undifferentiated state, are, at least theoretically, models of very early embryo, and not at all to do with the adult nervous system. Protocols are being developed and getting better all the time to differentiate those cells to adult phenotypes that really are truly representing what's in the adult, but they're not there completely, and we need to make sure that we're understanding the difference between development and adult cell types. This is a point that I don't really need to make too much, but just that the correlation between mouse and human data has gotten us into a lot of trouble before. Okay? Much of what we know about embryology comes from studies in mice. And as we're learning more now that we have human embryonic stem cells to compare to mouse embryonic stem cells or real embryos, some of the things that you would think would be absolutely conserved in evolution, completely not. It's amazing how diverse just mouse and human are, let alone software or anything else. And some examples of this from early developmental studies. DNA methylation patterns of the really early embryo are completely different from mouse and human. DNA repair mechanisms, which sets of molecules you have to repair your DNA, a process that goes on all the time, completely different in the two systems. Same thing with genes involved in drug metabolism, doing more 50 systems and all that. It's a whole different ballgame, which is good and bad, right? It's good in the sense that we now have human and embryonic stem cells to understand these things, but we still are not going to be able to get to potentially the same types of reasons we've had before. True early embryos in humans, because they're not accessible ethically and experimentally.
To be just to clarify,
Yep.
when you compare the mouse and human data, is it mouse embryonic stem cells to human embryonic stem cells, or early mouse embryos to early human embryos?
Early mouse embryos to early human embryos. And the same parallels have also been made in embryonic stem cells.
And so these studies, it's sort of stage matched.
Stage match, that's exactly right. I should apologize from the beginning. These aren't in any particular order, so that's probably going to be jumping around as I go through my list. Well, another important thing to think about is metabolizing the toxins in drugs. So, BPA or whatever molecule you might be interested in studying, you put that in a person, in a physiologic situation, who knows how many different metabolites of that get generated. All of those might have potential toxicities that nobody's thinking about. But I should say, people are thinking about that, but it's an important point when you think about these studies. It's not just about taking a dish of cells and putting in your particular compound, looking at the effect. You have to understand it in a true organism because of the first pass metabolism, liver changes all these things. There's all sorts of stuff that goes on. It's not about individual molecules. It's about, call it a tree of metabolites of each individual thing, which may or may not exist depending on the thing. Another important issue is so-called three-dimensional environment. those pictures I showed you of embryonic stem cells sitting on a dish, this is not at all physiologic, right? The three-dimensional microenvironment that cells live in has extremely important implications for the cell biology of that cell. They're sitting in the extracellular matrix, they're connected to different cell types, the muscle cell is sitting on a basement membrane, whatever it might be. Those kind of things are difficult to model in vitro and totally can't be ignored if you're going to start to develop the types of assays that we're leading up to. Quality of the cell is also very important. I know that the drug companies are very interested in developing high throughput methodologies to do this. You might take a little plastic plate. In the old days, it was a 96 well plate. They now have one with little teeny holes in 384, I think it is, and you can put stem cells in every single well, have them all nice and growing, put 384 different drug candidates on that, have some robot that moves it over to the next stage and it reads cells dead or alive, or potentially you could do gene arrays on every single well, whatever you wanted. If you're going to do something like that, you have to have very careful quality control that what you actually think you have is what you have over and over and over again, or you're going to lead yourself down blind out. Passage number, also important. This gets to the whole idea of epigenetic changes, right? The longer cells live in a dish, and this includes embryonic stem cells, the more they accumulate. Mutations isn't exactly the right word, but changes. And if you have a passage, if you have cells that have been passaged or moved from dish to dish 5 times, frozen, compared to 50 times, even though it's the same genetic background, the same cell line, chances are that over time there's going to be changes. You've got to be very, very well aware of that. How to construct assays and interpret endpoints. What are you actually going to look at? So this gets back to these isoprotocols. The cells look like hell and they died. That's not really defining a toxicity, right? And there could be changes that you don't see in those cells because they didn't die according to the endpoint of these protocols. We need to get much more careful about what our endpoints are going to be and realize that the endpoints are going to be complex. Gender specific, something else Eric also alluded to. So if you've got a girl stem cell line versus a boy stem cell line, there's no reason to think they're going to act the same in response to a particular exposure. Doses, I already sort of talked about this. You need to make sure doses are relevant and that you're actually thinking about local tissue concentration. There's all sorts of molecules that you can measure in blood, and there'll be one concentration, but then you measure it in a tissue where it actually means something to a cell, completely different and potentially much higher. Niches, this is a fancy word to talk about the local environment that a particular cell lives in. We have invented an in vitro niche that supports embryonic stem cell lines. And is that a real niche? Is that a niche or an environment that exists in early development? Probably not. Okay? These cells do represent cells of the inner cell mass, but whether they're exactly the same, they probably aren't. It's because you can't duplicate a niche or a particular point in time in vitro. And again, you just have to be careful in designing experiments about making sure this is at least thought about. And one good example of this is oxygen concentrations. In early pregnancy, in that period that Eric was talking about, the embryonic period, week 4 to 8 is when all these organs are forming, and after about week 10, it's all about growth and maturation. During that time, there is a relatively low oxygen concentration, hypoxia, and it's physiologic. It's supposed to be that way. So all the responses that cells have to oxygen are different in that stage of development than they are later when you get the placenta cranking blood and all this kind of stuff. You've got to think about that. Okay, and this meaningful endpoints point, I think. Yeah, so let me just give credit. My colleague Susan Fisher and our research group, these are sort of people that generated a lot of that nicotine data, so I wanted to make sure that put their name on the slide. I hope this helped frame the discussion. I hope it's going the right way. Thank you very much. I'm happy to take questions.
Actually, so you mentioned, so nicotine interacts with what you call, is it the nicotine receptor? Is that what you said?
Yeah, so
what's the normal function of the receptor?
Acetylcholine receptor.
I see. And
you got them all over your body.
I see.
Yep, and there's nicotinic and muscarinic.
Mm-hmm. And the pathways that you see upregulated, how does it compare to the pathways that would be upregulated if the receptor was hit by its natural, by its endogenous?
No, not at all. Not at all. And, you know, we haven't gone back to the second, but nicotine and acetylcholine are different molecules, and even though they're acting through the same receptor, the signaling pathways, the kinetics, all that stuff are going to be completely different. And toxicity is different from neuromuscular junction transmission, thank God.
But you're not hitting the muscarinic receptor, so.
Pardon me?
You're not hitting the muscarinic receptors.
Right. Nicotine is not an agonist for muscarinic receptors, that's true.
We're in that.
Okay.